Well, hello everyone. I want to welcome you to our Young Investigators webinar series sponsored by the Westchester Biotech Project. We are a fairly new organization. We just launched in the spring of 2017 and are putting together some really wonderful programs that are really about bringing together researchers, engineers, and data scientists in the context of our entire biotech cluster in order to enhance biomedical research. These are some of the different programs that we have going on, and they're all developed through conversation with our community. I'm not going to go into detail about these, but it will give you a sense of what we're about, and we'll be very happy to talk to you at length about any of them. My name is Joanne Geer, and I'm the Executive Director. I'm going to mention our partner and co-founder, Michael Welling, who is also with us today. And I cannot go further without thanking our community partners. These are organizations, companies, universities that are helping us to make these programs possible for you. During the course of the webinar, feel free to enter a question in the chat box, and we'll be addressing them at the end of the presentation, but feel free to enter them at any time. I'm going to hand it over now to Dr. Eileen Geoghegan from Albert Einstein, and she will introduce our speaker. Eileen, I'm going to make you, there you go.
Hi, Joanne, thanks. So today we have Rick Huntress from the Jackson Labs. He is the Director of Business Development, and we're very grateful to have him today. So Rick works directly with researchers, sponsors, engineers, and data scientists to design and execute early stage drug efficacy evaluations. And I've seen the slides. I'm really excited to hear what he has to say today. So Rick.
Thank you, Eileen, and welcome everyone who's set aside time today. I appreciate it. I wanted to just quickly ask, since we're all remote, are the slides presenting? Do you see the first slide there today?
They're beautiful. Looks great.
Okay, very good. So the goal for today is we have multiple goals. The first is to maybe look at this topic of generating data with the purpose of soliciting investment in maybe a slightly different paradigm. And that is to understand the perspective of the investors who are making these decisions, and how to develop data in your laboratories that's going to be aligned with the expectations of the market. One of the things you'll note is I'm going to be integrating business terms into this discussion. And I think it's important to understand the language of business, especially when you're coming from academia and you're interacting with business. Greater understanding always results when we're using a common language. And the last topic we'll discuss, and we'll go into some detail on, is risk reduction. I'm not going to describe what risk I'm referring to just yet. We'll wait till we get there. The last two comments I wanted to make as we start is please do pose questions. As Joanne referenced, those will go into our chat list, and we'll discuss them at the end. And I also want to be clear, this is one perspective, and it's difficult in terms when you're describing scientific invention and business to come up with one set of rules that apply. So there is no one solution. Some of the folks on this call may well have different experiences and different perspectives. We'll certainly respect those, and if you want to discuss those at the end, we can do that as well. So as we go through the more detailed goals for today, we're going to learn about the marketplace of in vivo data. When you're soliciting investment in your discovery, what is the marketplace you're working in? Are you selling a product, or are you selling an idea? Who are the buyers? Who's the person who's investing, or the organization that's investing? And who are you competing against? I use these terms marketplace, and all of the rules that apply to any market, whether you're selling cars or ideas, apply here. So let's talk about ideas and inventions. You're selling an idea. And the reality is you're probably further along than that. You've generated data. If you're at the point where you're interested in talking with outside investors or biotechs, it's likely there's data that you find compelling. It could even be a drug that's ready for IND and in the clinic, but you probably aren't that far along. If you were, it's likely there'd be investors involved already. So what I've done here is tried to bookend to some degree, what's the playing field that we're in? It's not simply an idea. It's not a fully fledged product. It's somewhere in between. So rather than ask yourself a question about the science that you're executing, this is a theme you'll see throughout the discussion today. It's what don't you know about the marketplace you're planning to enter? I often find that there is a discussion around the difference between intelligence and knowledge. Anyone on this discussion today, certainly we would consider to be intelligent, but if you're not familiar with the marketplace, you may have assumptions about how it works or functions. And like anything, if you spend some time understanding that marketplace where you're going to try to do business, you'll have a better experience and a better outcome. So what are we selling? Well, Rick, we're selling a drug. Is this a complicated discussion? Was the hypothesis sound, and does the experimental data support that hypothesis? These are the usual questions, and this is how scientists in particular look at their own data and the discussions that they're having. What's the risk that you have when you try to engage with an investor or a biotech? And what risk does the buyer have? Are they the same? This is to some extent, it's not a rhetorical question. In the simplest example, an investigator risks very little when they solicit investment. There's often a CDA that's required before a discussion takes place. There's a sharing of information, and if the investor or the buyer isn't particularly interested, the scientist goes back, the investigator goes back and continues their work. So there's very little risk. But what is the buyer risking? The obvious answer to that is whatever they're planning to pay or whatever's being asked in terms of a price for the idea or the invention. And also their time. When you work for either a biotech, particularly a startup, a VC firm that's providing seed money, time is valuable. There's only a certain number of projects or investments they can make in a year. They need those projects and investments to move forward. And if something doesn't, they can't get that time back. And in most cases, they really can't get their money back either. So I like to ask this question. Again, this is part of the theme for today, is trying to change the paradigm under which you review or look at your own work. Are you selling a drug or are you selling risk? I would put forth the proposition, and you'll see as we go through here, that what you're selling is risk, or you're selling an opportunity with risk, might be a more precise way to phrase it. So I'll ask the question of the audience here, do you understand the risk that your potential buyer is going to be exposed to? So again, going back to this original idea, what don't you know about the marketplace you're trying to enter? We've talked about the buyers can be biotech companies, they can be VC organizations. There are other types of buyers. Your own internal tech transfer department. This is a group that is typically looking to leverage discoveries of the institution to generate revenue to support additional research. They don't have an infinite bandwidth. And so even internally, you can view your own tech transfer groups as potential buyers. And as with any situation, if you view somebody as a buyer, it's going to change how you potentially interact with them. What criteria do these different groups use when they make investments or they buy products or discoveries? And are they using the same criteria? I would put forth the proposition right now that likely they aren't. And again, as you start to think about these questions, you come to the obvious conclusion. If you have different marketplaces for your ideas that use different criteria to evaluate them, customizing what you're presenting so that it's aligned with the organization you're talking to is a great strategy in terms of improving the likelihood that someone's going to be excited about your idea. Who are you competing against? Everyone is familiar with the dynamic of publish or perish, publish before your competitors do. There's always a certain amount of scientific irony in that in any niche, your competitors are also the collaborators with whom you share information when you go to scientific meetings. It's their papers that you might read to help you move your own ideas along, and the goal is to publish before they do. Well, in the same way, those discoveries that are being made at other academic institutions are going to be put forward into the marketplace as potential products or drugs. Many small biotechs, startups, particularly incubators, may well be working on similar ideas. And so again, if you make the assumption that there's more than one kind of organization you might sell to, there's also going to be more than one type of seller that you may be competing against. I realize some of these things are obvious, but part of the goal here for today is to, again, introduce this perspective so that you incorporate it into how you approach making your research available. So we're going to refine this a little bit more. So I'm going to talk about the risk. And again, going back to this idea of what are you selling and what are they buying? How do we make this fit together? And I'll just do a quick check-in with Eileen in terms of the pace and everything, the transition with the slides. Are we going okay?
Yeah, everything's working great.
Great, thank you. So what are investors buying? This table here, and there's lots of different versions of it, is one that I think almost everyone is familiar with. And whether you're going to use the number 200 million or 2 billion, we all know that it takes a long time and it's very expensive to get drugs into the marketplace. In many cases, the buyers that you might be selling a discovery to are not going to be the folks who take a drug all the way through this process. They may move a drug from the laboratory to IND approval and then package that asset and sell it to a biotech or a pharma. They may get it into phase 1 and there's an acquisition. But at the simplest level, there's a commitment of years and millions, if not billions of dollars. And so your invention is going to cost your buyer a lot more than just what they pay you for it. And not every drug makes it. And the reasons that they fail are wide and varied. However, do keep in mind that the folks who are buying these discoveries and moving them through IND, phase 1, and ultimately into the marketplace are intimately familiar with why drugs fail. And their programs are designed to try to mitigate this risk. I highlighted phase 2 here because phase 2 is typically a more complex evaluation phase than phase 1. You can see drugs fail, a third of them fail because they don't work. 22% might fail due to safety, which was not something that surfaced in a phase 1. And then you start to have other reasons why drugs might not move forward. And some of these seem counterintuitive. The rationalization of the company portfolio. If a company licensed an asset, they're purchased, the new ownership goes in a different direction, they're going to focus on neurodegenerative and not metabolic disorders, they might choose to simply abandon that project. And then there are other reasons why drugs may fail. The point here is not to highlight anyone specifically, but again, if you understand how your potential buyers or investors are evaluating your technology, you can start to address what might be their concerns in your own planning. So what percentage, based on the prior table, what percentage of compounds that enter a phase 1 trial go on to approval? It's only about 8%. What's the risk that investors have before they even get to an IND? only 40% of IND applications are approved. So the statistics here are challenging. We're talking about somewhere in the neighborhood of 3 or 4% of the drugs that get IND approval go onto the marketplace. If you're a VC company and you're buying pre-IND assets to advance to the clinic, to simply get one drug all the way to the clinic, you're going to have to buy 30, 40, 50 different inventions or discoveries to get one that wins. The typical return on investment in a VC portfolio can be anywhere from 30 to 40%. Folks don't get involved in venture funding for a 10% return. You can get a 10% return on your investment using a good broker, and if you're going to go for something more aggressive, you need to have a higher rate of return than that. Again, using these business terms to understand what are investors looking for. If they're looking for a 30 to 40% return on their investment, they're going to be looking at potentially hundreds of inventions. And I'll ask the question, how much would you bet on a 30 to 1 long shot? And this is at the IND approval stage. And what they're betting is a lot more than what they're paying to license your technology, because they have to then move that technology forward. So this is an important part of the discussion, and I'm going to transition here to talking about the business landscape to talking about research. And in particular, I'm going to focus on in vivo preclinical research. During my introduction, everyone knows I work for the Jackson Laboratory. I have been involved in preclinical evaluation of drugs since the late 1980s. And one of the trends that I've seen, particularly in the last 3 to 4 years, is this challenge around the predictive value of preclinical research. And this plays out in 2 ways. First is that what happens in an animal model system doesn't always happen in a human. But more concerning than that right now is the quality of the information that's in the public domain. Peer-reviewed published journals are considered the gold standard for scientific research. And yet, as you guys can see with my first bullet, 50%, this is a review that was conducted by the NIH, 50% of peer-reviewed published in vivo animal data and studies are unrepeatable. I'm going to stop and just let folks think about that for a minute. 50% of the data that's being shared publicly, that's been reviewed by experts and in some cases even competitors of the lab that generate it, is unrepeatable. If you're an investor in a pre-IND landscape, there's a 50-50 chance that the data set you're looking at is something that can't be recapitulated. So that 30 to 1 long shot we talked about earlier is now a 60 to 1 long shot based on whether or not the data itself can be reproduced. I expect to have a few questions around this when we get to the chat. And why this data is unrepeatable varies. This was a study done by Nature Reviews looking at comparing different publications around similar phenomenon. And what they found was even in the published literature, there are inconsistencies in the data. One of the interesting conversations we have at the Jackson Laboratory with the transgenic animals that we work with is we'll often get a call from a researcher who has a publication showing that when you treat a particular transgenic animal, you see an increase in expression or an increase in a particular phenotype that might be evaluated. In most cases, we can find another publication that demonstrates the opposite phenomenon. That's a very challenging landscape in which to try to select and understand what animal models you might want to use. And this phenomenon is not unique to published data. This is not a new topic in terms of, for those of us who are a little older. It's been in the mainstream press, and it's been addressed at the NIH. In the last 2 years, the NIH has significantly increased its guidance to researchers on how to execute work that's reproducible. It's a very detailed document, and if anyone's interested, I can refer folks to some of the directives that the NIH has put together. It has all of the things you would expect something like this to include, and it talks about study size, end selection, the use of statistics. I'm not going to necessarily do a deep dive on this, but simply wanted to provide the perspective that even within the research community, there is a discussion taking place about how to provide better work. The last comment I'll make before I move on here is we're all talking about the publication of positive findings. When you include that there is a real dearth of publication around negative findings, that complicates this issue. If something doesn't work, it's not published. Oftentimes, work is repeated multiple times by groups that aren't aware that things have already been evaluated. So those are the two challenges, is better reproducibility of data, but also finding ways to bring negative data into the scientific dialogue. Okay, so transitioning back to the business discussion, a buyer has to evaluate both risk and reward, not just the cost of the risk, but the likelihood. And in terms of the amount of revenue or reward, also the probability of that success. And if you know that the folks who are going to be evaluating your inventions are going to be doing that kind of assessment, ask yourself the questions. What have you done to reduce their risk? What have you done to reduce the costs associated with the risks they have to analyze? And what I mean by that is there may be some things that are relatively inexpensive for you to validate that might save somebody else down the road a good bit of money. And this will increase the probability of success, both in terms of the discovery you've made, but also in terms of your ability to market that discovery and get investment. And I'll mention one other thing here. Not only should you have thought about this, but actually state it out loud when you're working with the investment community. You talk to them about your discovery, but also explain to them, hey, I understand your landscape. I understand you're concerned about some of these risks. Let me talk to you a little bit about how we've addressed that in the work that we've put together. Because most sellers focus on the upside. Hey, if you get this drug to market, you're going to make billions. Or if you get a biotech to buy you out, you're going to make millions. But they really don't acknowledge the risk side of that equation. If you demonstrate to your investor that you understand the risk and that you've implemented some risk mitigation into your own programs, you'll see it helps with traction. And that's simply what I'm proposing here is if you acknowledge that risk and took action, you should expect it to have an impact. So going back to the theme, we're going to go back a little bit onto the science side again and talk about what do we mean by risk reduction? What are some of the things that when you get down into the weeds, folks are interested about? I will admit, and I apologize for the bias here, because my background has been preclinical research, I'm going to focus on that particular area. The NIH and other data sets that I showed you in terms of preclinical, it is the world that I live in. I don't want to provide the perspective that there aren't concerns outside of that, or there aren't things you can do outside of your preclinical, but this is what's I'm in the best position to describe to you today. So we'll talk about translational models. What are the models that are most relevant to the human condition? How are you incorporating biomarkers into your program? And I'll explain what I mean by that. And how are you handling statistics, both in terms of providing strong justification for your conclusions, but also being able to represent that in a mathematical and quantitative way. And then lastly, how do you show reproducibility and transportability of a discovery? So I'm going to say something that when I talk to clients and collaborators of the Jackson Laboratory, they often find confusing. I've stated it here in a softer way. I'll say it a little harder way. An animal model is not a good model for humans. If they were, we'd be a lot further along. The question I often get when I'm working with a new organization or someone who's new to the preclinical space is, Rick, what's your best animal model for this condition? What's your best animal model for Alzheimer's disease? The answer is there isn't one. And if you try to identify an animal model that is the best in terms of how disease is induced, in terms of the genetics, or in terms of things that might be added into it, you're going to have a rather naive approach that doesn't answer the questions that many investors and biotechs might have. And it's for the following reason. So when we talk about induction of disease, in colitis, DSS and TNBS are two chemicals that are commonly used to induce colitis in mice and other animals. It's referred to as a barrier disruption model. It does not have an immune system component to it. What it does is it strips away the mucosa, it damages the epithelial cells, and punches holes that allow proteins, materials, and blood and immune cells to infiltrate that epithelial, as well as bacteria and other things. It's an excellent model if you're trying to treat the symptoms of colitis, but if you're trying to mitigate or impact the causes of colitis, it's completely the wrong approach. And yet probably 75% of the requests that we get for modeling colitis are these barrier disruption models. The primary reason for that is if you look in the published data, animal models for colitis, probably 90 to 95% of the references you get are going to be for DSS and TNBS. So the weight of historical data focuses on these. If you're new to this space, you're going to assume that, well, this must be the gold standard. But when you dig into the biology of the disease, this particular induction does not mirror that biology. Another example is genetic models. There are many, many transgenic mice. We have transgenic rats. There are some examples of goats and sheep. And oftentimes we're putting human genes into these animals. But in some cases, the genetic modifications might have been introduced for a different reason. Early on in genetic modification, things like E and U were used to make point mutations throughout the genome. There's also animal models that have spontaneous mutations. One example I use here is leptin deficient mice. For those of you involved in metabolic research, DB mice and OB mice. These animals get enormously obese. They get to the point where they're so fat they can't walk, and we have to put their food on the bottom of their cages. The biological pathway that's mediating this is the leptin deficiency. And if you're looking at strategies to either remove fat or reduce the deposition of fat, it's a great model. But it's not how people get fat. Less than 1% of the human population, it's probably less than 1/10 of 1%, are leptin deficient. And yet again, 50 to 60% of the type 2 diabetes data that's published is in this mouse model. So given that the mechanism by which these animals have altered cholesterol and sugar glucose metabolism and obesity has nothing to do with diet and exercise, why would you expect this model to work? This is really indicative of, again, the technologies that were available in the prior century, the end of the prior century, really. I'm getting old when we can describe it that way. And there's a lot of historical data. I think we're going to have a little chat at the end about, how do you address this issue when a newer translational model doesn't have as much historical data behind it. That'll be a great topic for the end. The last example I want to use, and I'll move on from this slide, tumor cell lines. They grow well in immunodeficient mice. These are often referred to as xenotransplants, where you're putting a human tumor into a mouse. Because the immune system of that mouse has been repressed, those tumors will grow. And there's an enormous body of research showing drugs that can impact the tumor growth of these cell lines in mice. The challenge has been that in 40 years, very little discovery was leveraged. The scientific phenomenon was fabulous and exciting. You could put a human tumor in a mouse and it grows. When you treat it with the drug, it shrinks. But in the end, when you looked at the data that was generated and then applied it to the clinic, human tumors didn't respond in the same way in humans. And yet this was the gold standard for over 30 years. Just in the last 3 years, the National Cancer Institute announced that it is not really doing away with, but de-emphasizing an evaluation panel they referred to as the NCI 60 panel. This is a panel of 60 tumor cell lines. And before any cancer drug could go into clinical trial, you had to show data against those 60 cell lines. They're now removing that requirement. And they're moving on to a newer platform, which is referred to as patient-derived xenografts, which is PDX. So these are tumors that are taken directly from patients and propagated in mice without ever having touched plastic. Our basic assumption is that these are more translationally relevant than cell lines, which are grown in plastic. But at this point, we're still confirming that. So what does this mean in terms of trying to move your product forward? So I have a little case study here. This is a real conversation that I had with a biotech company. It was a clinical chemistry group, combinatorial chemistry group, that contacted me to say, we have a drug that we want to get into the clinic, and we want JAX to run an IND enabling study with it. And I was very excited because that's exactly the kind of biotech I like to do business with. It's where I'm focused. And I was asking some very simple questions. Well, has the drug ever been dosed in a live animal? And I was surprised when they said no. And I said, okay, well, what kind of tumor type or what mutation, what biomarker is relevant to how your drug works? And they said, oh, we don't know how the drug works, and I'm not really sure what tumor type we should use. We had an academic who used a cell line in his lab and showed it shrinks that cell. I've got to go back and find out what kind of tumor that was. So you can see the picture that's developing here. I asked them, how are you planning to figure out what to dose the mice? And they said, we hadn't really gotten around to that. And then this last question, which is really the most important one, is when you talk about pharmacodynamics and pharmacokinetics, how are you going to ensure that the drug gets to the tumor? And they said, we hadn't even thought of that. Now, even after all of this, what they said to me at the end of it was, well, Rick, these are all good points, but could you really just quote the study for us? Oh, and by the way, we only want to use 4 mice per arm because our budget's really limited. If they produced a data set that showed a tumor shrunk and then tried to put this in front of an investment group, they're not going to get a response. And going back to all of the things we've discussed up to this point, it's very easy to sit back and say why. But the most important one for me is really the second point here is, how did you choose the tumor type? What's the biomarker? How does the drug work? If they don't understand how to select a patient and how their drug works, how is someone going to move this drug into the clinic? So the question I'll ask this group is, why do they think the bar is this low? I'm not going to criticize their thought process. These were chemists. They were in new places. They didn't understand the market. But this is what happens when you don't understand the marketplace that you're trying to enter. If you're willing to spend somebody else's money, you need to demonstrate that you understand the risks that are going to go into receiving that money and moving the compounds forward. I'll skip through this relatively quickly. This is just a quick reference slide on the cell line studies that I was describing. This is from 2014 in Science, so you can see it's been a couple of years old. So I referenced biomarkers. What is your biomarker? The FDA won't move a drug into the clinic if you don't have a biomarker. The biomarkers help you to identify what patients are going to get enrolled and how do you evaluate efficacy? How do you know a drug is working? And in an animal model, do you have a good surrogate marker? Is there a disease pathology that you can identify in the animal model that is similar to the human. In ALS research, it's often lifespan is a very simple marker that's used. Transgenic animal models of ALS get sick and they die in a relatively short period of time. They die for different reasons, some in terms of muscle wasting, other in terms of preservation of the nerves. But if you don't understand how your drug works and you don't know which model to pick, how are you going to demonstrate that your drug is relevant to the human condition? And that's the key, is how is your drug active in a way, or your discovery active in a way that's relevant to the human? Ask yourself that question about the model system you're in and about where you want the drug to eventually go. Isn't the answer to these questions all the same? It's typically not. You may have a readout in your animal model that is not relevant to the patient population. But if you can define that, you can demonstrate the value of the data you're generating to your investors. And an example of that is the following. This is a genetically engineered mouse. It's expressing a gene that's associated with familial ALS. So this is an inherited form of ALS. The mice develop symptoms consistent with ALS, but they don't get the entire disease. So there's neurodegeneration, but they don't necessarily have the same muscle wasting and loss of function. They do die early, though. And so many folks are using this model, and the readout they're using is survival. The problem is these animals don't die of ALS. They die of bowel obstructions due to constipation. It's almost laughable, and I've heard people laugh when you say this outside. But imagine spending 2 years and tens of thousands of dollars studying a mouse to make it live longer, only to find out that its cause of death was not ALS, but constipation. So why would you use this model if it doesn't die of ALS? The reason you use it is because it's a great system to look at neuronal and axon preservation. And if you think about preclinical studies as a way to validate the mode of action of your drug, how it impacts a system, I should say your discovery, rather than trying to create an experiment where you cure ALS in a mouse, you're going to come across as a much more sophisticated preclinical researcher who understands the strengths and weaknesses of the model systems you're working in. And this is a quick example. In the upper left, showing the mouse, they don't look particularly healthy. On the far right, the differences in myenteric plexus staining and peripheral axon counts showing over time the loss of these different neuronal types, and the survival curve in the lower left. But the obvious thing here in the middle is the CT scan showing the completely compacted large intestine of these animals that ultimately leads to their death. So on that cheery image, we'll move on to a quick discussion of statistics. This is another example, and we spend a lot of time talking about statistics because there's a very direct relationship between how many data points you have in a study and the cost of that study. So there's always a balance between how many and how much you can afford. So this is, again, a real conversation. I was asked if we can put 2 tumors on a mouse. Can you engraft tumors bilaterally? And the answer is, of course we can. And when we do that, it's principally to look at tumor crosstalk, cytokine signaling. And in some cases, clients are doing something like an intratumoral injection on one side, and they want to see if that impacts the tumor on the other side. When I asked them why they wanted to do bilateral tumors, they said to me, no, we figured we could double the statistical power of our study without having to use any more mice. So they were going to treat a mouse with a drug, and if both tumors shrank, they were going to count that as an N of 2. I doubt this would have gotten through peer review, but depending on how the experiment was described and the data that was presented, it might not even be obvious to a reviewer that that's how they had done it. So instead of trying to figure out how to improve your statistics from a financial perspective, how many of you are engaging with a biostatistician to help you plan your studies? Some folks are likely to bring in a biostatistician to review the study and help analyze the data, but what if you actually brought them in earlier to help you plan your studies? How would your study look different if you had a statistician? The most common mistakes around stats relate to the fact that, well, a lot of researchers have a basic understanding of statistics. They're not statisticians at heart. And so that familiarity, I don't want to say it breeds a contempt, but there's really a lack of understanding as to the real statistics behind studies. And instead of just looking at whether or not a phenomenon occurred, if you measure how big it is, so beyond the p-value, what I'm talking about here is what is the size of the effect? If you have something that has a large effect, you don't need as many mice or as many data points. If something has a small effect, you're going to need more. We see this often in behavioral studies. If you're trying to look at in an animal model system for someone on the autism spectrum, a model on the autism spectrum, for example, you might be looking to see a 20% reduction in repetitive grooming. That's a relatively tight window to have in what is can be a qualitative assessment, which is how much of the animal's grooming. You're going to need a large cohort to demonstrate that. But if you were looking to show the difference between 8 hours of grooming a day versus no grooming, meaning the behavior goes away completely, you can have a much smaller sample size. And we all get that intuitively, but there are mathematical models that can help you more precisely understand, based on the size of effect you're trying to validate, how many animals do you need to use in your study. So my last topic here is reproducibility. And this is, in some ways, the most important. And this is, again, kind of a mini case study. I've probably been presented with this 4 or 5 times in the last 5 years. It's an academic research laboratory that has an invention. They've attracted some interest from a VC or a biotech, but they've been asked to demonstrate that their work is reproducible at a third-party site. And it's very common that a CRO would be selected to do that. We are contacted by both the investor and the client, the academic lab that's going to provide us the materials and educate us on how to do it. And they put this following proposition on the table. We need to reproduce this work somewhere else, but candidly, it's very tricky. So what we want to do is we want to come to your site and we'll do the work at your site. And I have 2 responses to that. One is we don't allow outside parties to execute work on our campus. But the other is your investor really going to accept that? And when they go back, it turns out they're not. So if your technology is this finicky, is it the right time to be trying to get investment? Or should you be trying to understand why is it so important that one postdoc or that one research technician be the person who's executing that procedure? What can you do to improve the reproducibility? How many times was the study run in your lab? Was the study blinded? I would say less than 5% of the studies that we execute for clients are blinded at JAX. We have the ability to do it, but we don't do it. It's considered not necessary by many groups. However, we're seeing an increasing interest in that. Can you blind a study in your own lab? Can you set it up so that the graduate student or the postdoc doesn't know which set of mice they're evaluating? And then can the work be independently verified, either through a collaborative relationship or a CRO? And I think lastly, and this is really, I think, the most important one, can you demonstrate the phenomenon in more than one model system? And if you go back to where I started in terms of animal models, there is no one animal model that recapitulates a human disease. They recapitulate components of the biology of that disease. So if you have 2 or 3 animal models or 2 or 3 model systems, each demonstrating the phenomenon to impact a biological pathway, you've created a much more robust environment in which your technology can be evaluated. Really, to hammer this point home, ALS-TDI is a group It's a nonprofit foundation that directly sponsors research on drugs for ALS. They culled the published literature. They also received applications directly from researchers. And in this kind of aqua green color, what they're showing you is the average lifespan extension as a percentage of the different published reports. In each case, they sponsored that researcher and had the research repeated at an independent CRO. And in none of these cases were they able to recapitulate the original findings. Now, no one's implying here that the original research was done unethically, but clearly when it was done in an environment where processes were standardized across model systems and platforms, none of it could be reproduced. Going back to the original proposition I started with here, do you understand the risks that your potential investors are evaluating? Can you imagine being the next researcher to propose a study to ALS TDI after they'd finished this study? What kind of rigor is ALS TDI going to expect when they evaluate compounds going forward and when they sponsor research? So bringing it all together, and hopefully we're still on time. I've been enjoying this quite a bit so far. I look forward to the questions. So develop a de-risking strategy. Select multiple translationally relevant model systems that are aligned with the mode of action of your drug. Really make sure you understand how the animal model or the model system compares to the human condition. Make sure that the biomarkers that you're using to either demonstrate effectiveness or efficacy are aligned with that same MOA. Is your readout relevant to the way your drug works? Build the statistics into your design, not just into your analysis at the end. And find ways to reproduce findings both inside and outside of your lab, obviously within the constraints of the resources. And I'll go back to the proposition I've been making all along, which is when you understand how an investor makes their decisions, you can anticipate their wants in a very thoughtful way, and it will make your work stand out relative to others.